concentration, not only in the grain but also in the straw, and also soil mineral nitrogen. And we, at the end, we calculate some parameter of nitrogen efficiency. First of all, as a result, we can see that the 4 years was very different, with some years very, very cold, that in this one, but also with higher temperatures during the winter. But the more important difference were in the bottom graph, that are presenting the cumulative rainfall in the 4 different seasons. Yeah, you see that at the beginning we have a very high difference just during the 4 first months, but also at the end of the cropping system, moving from close to 200 millimeters to larger than 400 millimeters. So in the plant grow, analyzing this soil cover with the digital images, we can see that we have, yeah, for sure, 3 different patterns that correspond each of one to one sowing date. The main result is that the maturity of the wheat is quite close at the end of the season, mostly because of the increase of the temperature and the dry of the soil. But we can harvest in one week of difference. Yes, but the maximum reach of soil cover is get first in the October sowing, a smaller period of growing in the November, and a very small season for the February. And also we can see that there are a different growth respect to the nitrogen, being larger, this coverage in the treatment with the higher fertilization, and lower in the no fertilizer treatment. The results are very similar also in the different indices, but I'm not going to introduce in that because I don't have time. For the yield, we can see that there are an impact respect to the sowing date. In this case, you can see that all the results are cumulative for the 4 years, and you see that there are a large variability in the yields from year to year, and it's mostly due to the water limitation, for sure. But even when we have a response on the fertilizer rate, we can see that there is not a very huge amount, or even sometimes it's a reduction with the total fertilizer applied. So the half fertilizer treatments are working quite well. And if we see the biomass produced, the straw biomass produced, we see that the response is much clearer and is more similar to that we expected at the beginning. So most fertilizer treatments get more biomass. And combining that, we can see that the harvest index is representing these 2 different I don't know how to say, the development. So the biomass go by one side, but the yield go by another side. And this is telling us that probably this reduction in the nitrogen, coupled with the water deficiency that we are going to have at the end of the season, is trying to reduce a little bit the biomass produced, that is going to produce a very higher ETC at the end of the season. So if we have a little less straw, we are going to be able to go to the end with all the grains. So this nitrogen is going to keep our crops saying, no, don't go too fast, because you are not going to have water later. So it's a good solution. With respect to the nitrogen in the soil, we see something similar, and we see that there is a lot of lines, but the main thing is in the bottom of the graph, we found always the non-fertilizer treatment, but also the half fertilizer that has been sown in October and in November. So it's saying that our wheat, when we apply the fertilizer even before at the starting of the, the tillering, we see that this is able to absorb all the nitrogen. But if we apply in too much nitrogen perfect, or we are sowing in February, it's not having this time to take out this nitrogen, and it's going to produce also nitrogen with respect to the nitrogen use efficiency, we see similar results. So it's working very well in the 15 in the November sowing and also in the half fertilizer treatment. And for all the indexes work similarly. As conclusion, it's essential to find new crops strategies that allow farmers to adapt their crops to new climatic conditions. And adjusting sowing dates and nitrogen fertilization rates are two very easy and cost-effective alternatives for this adaptation, but also mitigation because we are applying half nitrogen. This yield and quality improvement can be achieved delaying with sowing, but also reducing half fertilizer. And the results are more or less constant along the 4 years, not so easy, but we can prevent some internal uncertainties. And also the use of proximal sensor could also affect or could also help to go further and try to correct this fertilization if we have a very good spring. Thank you.
So we do have some time for some questions. In Australia, in our heat systems, excess nitrogen is often used for an insurance just to make sure. Is this a similar barrier to the acceptance of results such as this design?
Sorry, the beginning, please.
So fertilizer nitrogen is often used as an insurance policy for the farmers think about this as an insurance mechanism. So if you are trying to convince the farmers to cut their fertilizer by half, there might be some other issues other than environment.
Yeah, it's similar in Spain. So people always are thinking in that what happened is if this year is going to be a record one, and if I don't apply fertilizer, I'm the silly one in the town that doesn't apply this amount. But yeah, that's a barrier that we have with the farmer. But when you put numbers and you say, okay, how many years during the last 10 years have you put the maximum fertilizer, and how much had you get in the yield? So perhaps you can lose 1 year, but you are gaining 9. So I think when you put money in this, they say, okay, I'm going to try at least in these 2 hectares and see what happens. And some of them get combined, not all of them, for sure. It's the way it is.
I noticed that you had that in these wondering how much impact do soil moisture on the results. So it's even
Yeah, no, for sure. The soil moisture is the key here. So yeah, the problem is that you never know what is going to happen in 1 week, 2 weeks, or even 1 month. So yeah, if you know I don't have measurement in this experiment because there are many treatments and I can't put a sensor in all of them. But yeah, no, water is key for sure. I don't hear you.
Oh, sorry.
I think you.
If you know how much moisture is in the soil at the start,
No, it's also a point because not all the years we have water enough at the beginning of the season. So, but anyway, the germination was not bad. So, yeah, some years we can have some losses, but I think it's not the point for germination. But yeah, the development at the beginning is key for sure. Some years we had almost no water, but the years that we don't have water, but most of the years it grow more or less fine at the beginning until February. But yeah, for sure, some year it's more important the water in the when we did some analysis, we found that it's more important the water in the spring than the water in the winter.
Thank you. Next speaker is in program changes. Speak if you're very confident. And the title of Helen's talk is Challenges and Prospects for Next Generation.
Thank you, Cameron. Yes, so unfortunately, Delhi was not able to present, and a lot of you would know that I'm not Delhi. Know Delhi very well. So this is some work that we're doing at the University of Melbourne, where we have a new research program, and I'm going to talk a little bit about that as we go through the talk. So it's hard to start a talk without saying this, even though we probably all know it, but for 100 years people have used nitrogen fertiliser to grow crops and feed the population, but the efficiency of that is very low. About 50%, some different numbers are coming around in the talk. And so the production is about 135 million tonnes per year. It uses 2% of the world's energy to produce that and costs about $500 billion US a year. Oh, that's better. Sorry, I'm not very good with microphones. But as has been mentioned in some other talks yesterday, the societal cost is much higher than the economic cost. So in our work at the University of Melbourne, we look at enhanced efficiency fertilisers, and these are fertilisers that change the way that nitrogen is released and made available to the plant. And they can be things that are coated with different things that cause different release. They can be urease inhibitors or nitrification inhibitors. And so the controlled release coatings are designed to just slow the release of the nutrient. The urease inhibitors inhibit the process of urea hydrolysis and therefore limit the emissions of ammonia, which we know from many of the talks is an important pollutant. The nitrification inhibitors inhibit the microbes involved in the first step of nitrification, and as a consequence reduce the production of nitrate, which reduces the risk of nitrate leaching and of denitrification losses of the greenhouse gas N2O and of N2 as well. And so at the moment there's essentially one commercially available urease inhibitor that's used worldwide, and there's a number of different nitrification inhibitors around the place. So in our team, Shuki Lam has done a meta-analysis and looked at how much these EEFs are effective in reducing nitrogen losses. And so just to make it a bit clearer, I'll show that the top is looking at ammonia volatilisation impact. So you can see anything that's a negative to the left of the line indicates a positive effect on the emissions. So urease inhibitors have a positive effect on ammonia volatilization. They reduce that. The nitrification inhibitors don't have an effect. They actually can increase it. The double inhibitors, which is a urease and nitrification inhibitor, have an effect, and also the controlled release fertilizers do. If we look at nitrous oxide emissions, the urease inhibitors here don't work, as we know. They can actually increase nitrous oxide emissions as well. Nitrification inhibitors work well, double inhibitors work well, controlled release fertilizers work pretty well. And if we look at nitrate leaching and runoff, then the nitrification inhibitors, by reducing nitrate, have an effect on that, as do the controlled release fertilizers and the double inhibitors to some extent. So the other thing I wanted to point out from this graph is that there's a great variation. So what we find, and I know a lot of people who've been coming to these conferences have been looking at these products, there's so much variation in how well they work. And this is due to differences in soil, differences in climate, differences in cropping systems. So we're interested in looking at what we're calling a next generation of enhanced efficiency fertilizers that will target some of these issues of variability in responses. And so what we're doing is we're bringing together a team of people who are involved in other areas. So it could be the chemists involved in some of the development of medical materials. We've got engineers as well working with us. And we're looking at integrating the traditional sort of soil plant science knowledge with synthetic chemistry, chemical engineering, and the plant soil microbiome. And we are also interested to think about the socioeconomic costs, which has also been talked about quite a bit in this conference so far. So this is what we're working on. It's called, this is ARC's Australian Research Council. It's an industrial transformation research hub for smart fertilisers, and we're about in the middle of this at the moment. It's funded by the government, and it's a collaboration between University of Melbourne, La Trobe University, Incitec Pivot Fertilisers, which is one of the larger fertiliser companies in Australia, and Elders Rural Services, which is like an extension team. Delhi is the director of this hub, and I'm the deputy director of this hub. So we have a number of different research themes in this hub. We're wanting to develop these more efficient, cost-effective as well, enhanced efficiency fertilisers, because that's a barrier to adoption often. We're aiming to improve the efficiency of nitrogen use by up to 20%, and we want to think about the societal costs and benefits. So the 5 themes are looking at development of engineered coatings, new types of inhibitors, plant-soil microbiome interactions. We have to validate and test everything with this agronomic and environmental social testing, and then maximising sector-wide value. And we have a prospective paper that's been written associated with that hub. So with the urease and nitrification inhibitors, there's a few different approaches being made. This is published information in the paper listed below. And this is, so it's changing, working with things that exist like NBPT and changing the radicals that are on that, and also looking at developing new types of urease inhibitors, also looking at current nitrification inhibitors, and also developing new ones. So there's a few different approaches going. I'm not going to go into all of these. There's lots of IP, and it's not my area of expertise developing these as well. The second area is looking at coatings, and this is one example of one of the types of coatings, and this is published in the paper listed below as well. And so this metal phenolic network hasn't been used in use on fertiliser products, and there's been a development of different ways of coating the fertilisers, what the release is looking like, and what that does in terms of supplying it to the plant. So a lot of these things are in the preliminary stages of assessment. And all of the coated products, we're aiming obviously for things that are biodegradable, environmentally friendly. Other fertiliser coatings we're looking at are ones that are associated with the plant. So it will either be that the coating has the plant signalling compounds in it, so it activates the microbes in the soil to provide nutrients to the plant, or it'll be ones where the coating responds to signals that are released by the plant when it's nitrogen stressed. But as mentioned in other talks, there is this issue of the cost. So we can do all these things, but there's the issue. We need to think about the cost of what all this nitrogen pollution is, so that people are motivated to actually become involved in developing and using these products. So in this slide here, you can see the costs associated with human health, ecosystems and climate. And you can also see on the right that ammonia is largely from agriculture, as is N2O and the runoff as well. So we've come up with this 3Ps policy, 3Ps concept, I guess, which has been led by Delhi and Emily Yang in our team. And this is about thinking about planet, profit and people. So it's about ensuring people have sufficient, safe and nutritious food, ensuring that farmers and producers are able to make profit from use of these fertilisers or use of nitrogen. And it's also looking at making sure that the planet is healthy and clean and sustainable for the long term in terms of the resources available. So at the moment, food is cheap. Farmers only really consider input costs and farm gate income, and the costs, societal costs, the cost of the planet, are not considered in decision making. And as with most things, the cost of prevention is much lower than the remediation costs. So if we can develop these products and there's a way of those being valued by society, that they can actually be used by people, then we'll get over the problems of having to clean up messes that are developed that's much more costly. And this is an example of the Great Barrier Reef here, which is very important, affected by the quite intense vegetable and sugarcane production in Queensland. So one of the other areas that we're looking at is getting a factual index. And I did go to another talk this morning that talked about a nitrogen footprint. So that's interesting, but obviously a lot of people are looking at this. So this is about, let's get some numbers around it. Let's get some scientific proof that these things are happening, that we are having positive impact, rather than just marketing things without necessarily having any strong science behind them. So similar to a washing machine water rating, a car environmental impact from the CO2, they're things that you can have numbers to validate that. Organic produce is something that's always been a little bit harder. Okay, so we want to develop this nitrogen index framework. I'll have to put my glasses on because I can't read all this, sorry. So looking at the supply chain data, remote sensing, field monitoring and modelling, and using the information that's collected from production systems to develop a nitrogen index for different types of food. And then this can be used to provide a credit to farmers so that they then are incentivised to work this way. There's a benefit for them. And it provides a way of labelling products so that consumers can then make a decision as to whether they want to do that, and farmers should be able to sell that at a market premium. So we're working on building these new evidence-based indexes. Here's an example of some work we did on celery, where we used the nitrification inhibitor DMPP, which reduced the nitrogen loss from that as N2O, and you can see that it had a nitrogen footprint of 4 stars, but when you can reduce that, we can obviously increase the star rating so that it's a way of marketing that to people. So this diagram shows that if we need to consider societal costs. So on the left-hand side, if we just consider costs and benefits in a traditional way, then agricultural products come up quite well. They're the heavier side of the agricultural products. Whereas if we then think about the societal costs, that's much bigger, and then the balance is less in favour of the agricultural products in terms of the nitrogen cost. And there's some work done by Bao Jingbu that looked at cost-benefit analysis by region, and this is about mitigating reactive nitrogen pollution. And just wanted to show this one because you can see that the positive benefits are above the line, and there's big impacts in these different regions of the world on health benefits, ecosystem services, and some yield benefits as well. So emphasising the fact that these benefits are things that we're increasingly considering more, that need to be considered more in policy. The nitrogen credit system is a way of working out how we can reduce this pollution, improve nitrogen use efficiency by sharing the responsibility so it doesn't all fall to the consumer or the farmer. It's shared by everybody along the supply chain. And then that gives a real financial incentive to use products that will reduce the environmental nitrogen footprint. So the take-home messages from the presentation are that, as the global population increases, there's going to be a requirement to put synthetic fertilisers out, we believe, to feed that population. Enhanced efficiency fertilisers have shown some promise, but there's been sort of little development and still unanswered reasons for the variation in what they look like. So it's hard to say, put a value on how much emissions reduction you could get from a particular product, for example. So we're looking at these new EEFs that are going to be more targeted. So we can think about soils, climates, regions, crops, and how they might work. And the societal benefits need to be considered much more in developing these fertilisers and policies. This slide just provides you with some context. If you wanted to look at Hub, the website is there. There's contacts for Delhi, myself, and ARC Hub Manager as well. Thank you.
Thank you, Helen. Very comprehensive. We've got time for one question. This one? I don't think I have a motion. How do you comment on looking at what the tracking was in the
Yes. Pardon? Oh, I think the question is, how would we go about actually tracking the nitrogen footprint along the whole value chain? Yeah. And I would say that actually this is not, I'm a soil scientist, so that's not 100% my area of expertise. But I guess there's a way of, aggregating the data that's available about, in the production system, I know you can see where all the losses are occurring there. So I think that would just be bringing those things in, I imagine, through what transport might do and where wastes might occur along that supply chain. But yeah, you could contact Delhi if you'd like some more information on that one.
One more. Now, how's the nitrogen credit system going to be funded?
Well, you've got to be aspirational sometimes, don't you? So I think we did used to have a nitrogen credit system in Australia that was kind of funded by the government, and it was going to be run through aggregators, and there were certain methodologies that were developed that farmers could use. I think there was only like 1 or 2 methodologies accepted. And so I guess I think the original plan was that it would be government funded, but I think it has to now be across, as I mentioned, across the responsibility has to be shared so that everybody's contributing, the consumer is contributing as well as the government's, I imagine, in that case. But it will be a bit complicated. Baojing has written a paper on that, actually. I'm not sure if I had the reference in the slides there. Yeah.
Well, thank you.
Okay, thank you.
We're going to move on to our next speaker, David Anders. The title of David's presentation is Nitrogen Management Practices in the US Core Sufficient to mitigate environmental nitrogen loss.
All right, well, thank you. Good afternoon. I would like to start by acknowledging my co-authors in this presentation, several graduate students and colleagues that work with me in the Department of Soil, Water, and Climate at the University of Minnesota. So today I'm kind of doing a synthesis of a lot of different papers, different projects that we have, and basically one objective overall, which is to evaluate the agronomic and environmental outcomes when we are using nitrogen-based management practices or MBMPs. And the idea here is to look at reducing nitrogen losses and also improving corn yield and net economic return. All of this work that I am presenting today is from Minnesota in the Midwest of the US. And in Minnesota, one of the great challenges that we are seeing right now is precipitation, in greater precipitation and more erratic distribution, along with warmer fall winter temperatures, which all of these kind of stack against us in trying to improve nitrogen management. They kind of work against us. And really and truly, when we look at this, I think climate change is really creating one of the greatest challenges in improving or showing achievements towards environmental stewardship. I think we are doing a lot of great things, but sometimes they get kind of overshadowed by some of the challenges that we see with climate and with the weather from growing season to growing season. So in all of these studies, we are conducting intensive simultaneous measurements of different nitrogen loss pathways, as well as bioavailable soil nitrogen, soil end supply, and the nitrogen that ends up in the plant, as well as in yield. What I will do today is I'll share, again, this is kind of a 30,000-foot view of a lot of this work. Talk a little bit about some different management practices that we can use and how they behave and what they do for us, and kind of look a little bit about the theory and then what we see in reality. And I'll start with nitrogen rate. So when we look at nitrogen rate, the theory will say that we increase corn yield to a certain point as we add nitrogen, and then it plateaus. And then in terms of nitrogen loss, we see fairly flat loss of nitrogen to about that optimum, and then really starts to skyrocket. Luckily, when we look at it in practice, we actually see something that follows pretty closely. This is just one example out of a 7-site-year study where we see this yield increase to an economic optimum and then a plateau after that in corn yield. We also see residual soil nitrogen in the fall, fairly flat from the economic optimum or below, and then a sharp increase after that. We also looked at residual nitrogen the following spring. We see that there's very little difference in nitrogen between these lower end rates to the economic optimum, but there is a substantial decline in nitrogen, residual nitrogen by the following spring. So the potential for loss, the potential for that nitrogen not to be necessarily plant available is substantial once you go above that economic optimum. Well, the theory and the reality work great in that. The challenge with nitrogen rate is determining what is the optimum nitrogen rate. We always talk about applying the right rate, but what is the right rate? And that's where it gets really challenged. This study here is over a 9-year period where we are looking at the economic optimum end rate and the yield obtained at those economic optimums. And you can see that the only thing that changed in here is the growing season. We basically, in these two, for example, we got the same yield with substantially different amounts of nitrogen needed to get to that point. In these others, we got substantially different yields, but we needed about the same amount of nitrogen to get to that point. And then if we look even farther, we look at a database that we have to determine the end rate calculator that we use with 120 site years of data, where if you look at the yield of the check plot with no nitrogen, you can yield between about 3 to 14 megagrams per hectare. So substantial range of variability there, and most of that is coming from nitrogen mineralization. Another thing that we are looking at is timing of nitrogen application. And so in theory, we know that rainfall patterns really determine the potential for nitrogen loss. We also know that corn needs nitrogen later in the season. And so if we can move the application from early in the season so that it's after this period of high potential for loss, but before the corn plant starts to take up a lot of nitrogen, maybe we can improve efficiency. In practice, though, time and again, we see that that doesn't really work out very well for us. In this particular study, this actually was across the Midwest, 49 site years of data, where we were looking at different nitrogen rates to get a response curve, comparing single pre-plant applications to two kinds of split applications. One where we apply 45 kilos pre-plant and then apply the rest at side dress, or 90 kilograms and then side dress. And 80 to 84% of the sites show no difference between the pre-plant versus the split application. So while in theory it looks like that would help us a lot, in practice we don't really see that working very well. We are also looking at source and timing of application, and in this particular study, we evaluated 4 different treatments. We have urea and ESN, which is a polymer-coated urea, as a pre-plant application. And then we had these 2 treatments were split apply, either with urea or ESN pre-plant, and then the rest of the nitrogen applied as urea with MBPT. The pre-plant treatments were broadcast incorporated, the side dress were just broadcast. And what we see here is the precipitation is really a big driver in what happens to nitrogen. You can see these bars represent the precipitation events, this axis. And while there was a nice reduction in nitrate leaching with ESN pre-plant compared to the other 3 treatments, you see that the pattern of loss is the same for all of the treatments. And they are associated with times of high rain events. And in here, I kind of marked those 5 that were in this particular year, but the same thing was observed in all of the years, where typically 3 to 5, 6 rain events drive the majority of nitrogen loss. And this is a big challenge because that's when we lose most of the nitrogen, but also we have such a big volume of water that it's really hard to do any kind of remediation with that water at that point. Then we look at ammonia volatilization for this same study. And here we have 2 years and 2 different stories. For the pre-plant treatments, they both had the same behavior where urea pre-plant lost more early in the season and then kind of plateaus. ESN pre-plant was fairly flat. But for the split application treatments, the difference of how well they performed had to do not so much with the time of application, but what happened after that application. In 2019, we had a very small amount of rain right after the application, just enough to solubilize urea and make perfect conditions for volatilization, and we saw substantial losses. Whereas in 2020, we had more precipitation, it got incorporated, and there was hardly any loss. And so again, precipitation is a big driver on nitrogen loss. And then looking at nitrous oxide emissions, kind of again, 3 different years, 3 different stories. In this first year in 2018, we had high loss potential at pre-plant and split timing. And you see that urea lost quite a bit, and the split treatments as well. ESN pre-plant was very low. In 2019, we only had high potential for nitrous oxide emissions happening in pre-plant time. And so you see urea really losing quite a bit, but the split applications did just as well as the pre-plant ESN. And then in 2020 was dry, and so we have pretty low potential both at pre-plant and split application timing. And so there was little loss, and all the treatments were pretty similar. And so if we put all of these losses combined for this study, pre-plant ESN reduced nitrogen loss by about 48%. So it's a substantial reduction, but even with that reduction that we get really excited about, it's still not zero. There is quite a bit of nitrogen that gets lost through either leaching, denitrification, and so we need to be aware of that. And so regardless of pest management practices, we know we are going to lose some nitrogen, and so we are looking at cover crops as one way to mitigate some of these losses. This is a 4-year study looking at the economic optimum end rate with no cover crop, rye cover crop, and then Acura, clover, living mulch. We saw that the rye did not really make any difference. Acura reduced the nitrate leaching substantially, but the challenge with Cura is that when we look at grain yield, it substantially reduced the yield. So it's really not a practical solution for farmers. When farmers see this, they are not going to try it. Plus, Cura clover is a much more difficult system to deal with. There's a lot more management involved in that production system. And then the other thing that we are looking at is soil drainage. People say that we have a very leaky system with nitrogen, and when we install tile drain to remove the excess water, we are making it leakier. But in reality, what we say in this study, where we are looking at pre-plant and split applications, this is just looking at the economic optimum end rate. So these were end rate studies. We found that consistently the undrained soils needed more nitrogen to produce the economic optimum yield and had lower nitrogen use efficiency, which is telling me that in a relative basis, we lose more nitrogen through denitrification in this system than we do when we tile drain and lose some nitrogen through leaching. So it's kind of this issue of pollution swapping. We also saw that pre-plant applications did very well compared to in drain soils, but in undrained soils where you have more potential for nitrogen loss, the split application was a better option. So with all of that, again, a very high synthesis of a lot of different projects. The key points here is that nitrogen-based management practices can improve production and environmental protection, but are insufficient to substantially reduce nitrogen loss. We see that the concepts don't always follow what we see in reality because there are just, too many unpredictable variables. And we see also that weather can very easily erase some of the benefits that we can gain through some of these best management practices. And so as we look forward to how to deal with this, I think we need to use best management practices, expect nitrogen loss, and then figure a way to deal with that nitrogen loss. Try to recapture as much of that as we can. And then a very important question, I think, is to ask ourselves, is the environmental benefit worth it? And how much does it cost? Somebody has to pay for that cost. And like Helen said, it's not fair to put it all on the farmer or all on the society, but having those values can help us deal with some of these things. And then targeting the EONR is probably our best option. When we apply less than what we need, we don't really have a benefit for the environment and we reduce production. And so the best game will be when we are over applying. If we can bring farmers to apply the right rate, which again is difficult to predict, but moving it down to where it's a reasonable amount, that's where we have the biggest potential for gains. So with that, I will stop. Thank all the sponsors to the work that I presented today.
Thank you, Fred. I'm sure we'll ask some questions. Thank you. I'm interested in one of the slides where you showed optimum nitrogen rate is changing year to year. So, what is the solution to farmer? Do they have to, how do they manage it? And how do they determine agronomically or economically optimum nitrogen rate? And considering that you need 4 or 5 rates of nitrogen, and how do you do it before planting?
Yeah, it is a very difficult thing to address, and it almost sounds like a brute approach. But I think one of the things that we are doing in a lot of the states in the Midwest is we have a database of corn nitrogen responses, and we look at how much nitrogen it takes. We estimate the economic optimum, and we have a larger database that allows you to say, okay, with reasonable assurance, this is how much nitrogen you will need. But we know that there is quite a bit of a range. And so with that value, at least you can, that to me is like the starting point. Use that nitrogen rate out of a large database, because again, there's a lot of variability, And yes, we are managing on the average when we do that, but that's better than just saying apply whatever you feel you need. And then more progressive farmers can use that value and then evaluate it in their own farms and decide, do I need to go up or down from there, depending on the conditions.
Thank you so much.
Thank you.
To our last speaker, full speaker, I should say, at 15 minutes. That's it. Australian. He's going to change society. Thank you.
Well, thanks everyone for staying around and listening to my talk. I'll be very quick because I got quite a few slides here. So, a bit of a change in pace, and this is some work that we've been working on for at least 20 years collectively, but a big push in the last 3 or 4 years. Just to acknowledge a range of people that have been involved in this and our funding agencies that have funded the work. I'm going to go through a journey of how we identify these classes of ammonium facilitator proteins called AMFs. So we first identified a transcription factor many years ago called BIHLHM1, which was expressed in soybean nodules. And it basically is expressed as the nodule starts to fix nitrogen. And if you disrupt that transcription factor, you disrupt the nodule symbiosis quite significantly. And when you express that transcription factor in a yeast strain, it was able to identify a novel membrane transport protein called AMF, which allowed the yeast strain to take up methylammonium, which is an ammonium analog. And one of the interesting things about this AMF is that it's co-localized with this transcription factor in all sequenced dicot plant species around the world. So AMF1 is an ammonium transport protein. It can transport methylammonium, transport N15 labeled ammonia. Tends to work at higher concentrations of ammonia. This is an expression of both yeast and soybean AMF proteins. It's a transmembrane protein, obviously. They're linked or homologous to DHA2 proton drug antiporter proteins, common in bacterial systems, and as well as fungal systems. And so generally, there's a proton antiport mechanism that operates with these proteins, and they transport a range of different things called from amino acids to siderophores, boron, and potassium, depending on the type of protein. So we've landed on an ammonium proton antiporter in this study. So these things are present in all plant species, generally present in just about every cell type at a very low level of expression. And so we've done some preliminary work in soybean, and we've been able to identify one of the AMFs in soybean nodules called AMF 1.3. It does transport ammonium and methylammonium, very similar to what we saw in yeast, and is probably a bit more active in methylammonium transport than the yeast version was. And this AMF3 in soybean is mainly expressed in the outer cortical cell types, often associated with vascular tissues. And it does interact with the transcription factor, though. So this transcription factor we initially identified binds to the promoter of these AMF proteins to transcriptionally activate and turn it on. And if you disrupt this in soybean, it doesn't kill the nodule, but it just slows its development, and then it starts to senesce earlier. And you see this both in soybean and medicago. But it also disrupts the AM mycorrhizal symbiosis, and we just published that recently. We're interested in studying the structure function of what these proteins do, and so we've used the model system Arabidopsis, which has 3 AMF proteins present in the background of this plant. And when you look at the expression pattern of these AMFs, they tend to be expressed in older tissues, particularly AMF2, which you start to see its expression dominate as the plant cells get a bit older, but it's generally expressed in all 3 different cell types. And that also includes AMF1 and AMF3. If you look at a GUS-labeled expression pattern for these AMF tissues in Arabidopsis, often you'll see them in vascular tissues, of course, AMF2 is generally the predominant protein or gene that's expressed in a range of tissues from shoots, leaves, petioles, roots, and so on. Look at where these proteins are localized, because it's really important to put them into context of how they would be involved in ammonium transport from plants. If you look at the surface cells in Nicotiana Bethanimania, basically you'll see expression of AMF1 in the endoplasmic reticulum. AMF2 is a tonoplast specifically localized protein, and AMF3 is localized in the plasma membrane. And you can go flipped into an Arabidopsis cell expression system using mesophyll cells, for example, and get the same sort of phenotype. And you can track that also in yeast cells. So if you actually overexpress these proteins in yeast, they'll be predetermined targets to different membrane types. So the ER, tonoplast, and PM stand out as targets for AMF1, 2, and 3 respectively. So we've done quite a bit of biophysical work on these proteins. There's still more work to be done, but when I was in Adelaide before I moved to University of Sydney, we spent a bit of time looking at stop flow spectroscopy in the lab of Steve Tarman's group. And we were able to identify that all 3 of these proteins in Arabidopsis do facilitate the movement of ammonium into yeast cells. This is shown by a swelling of the vesicle in response to a transient pulse of ammonium. And you can use radioactive tracers such as methylammonium and look at the quantification of flux of isotope into yeast cells. AMF2 shows quite a significant response to methylammonium and AMF3 as well, but AMF1 was really quite active as a methylammonium transport protein. So I'm going to switch here a bit because you know that ammonium can be quite toxic to most plants, and that nitrate and potassium are often antagonists to ammonia and actually can recover the ammonia toxicity. So this is some work from Jan Schuring's group back in 2007, 2008, showing recovery of ammonium toxicity in cucumber with either the presence of equal molar concentrations of nitrate with ammonia or potassium. So we've been trying to understand what these proteins do, and we've been using the yeast model system to actually get a better handle of how they function. We know that yeast cells take up ammonium using high-affinity ammonium transporters called MEPs. There's 3 of them, and often that ammonium will be partitioned into the vacuole to actually acid trap it as ammonium, to remove that ammonium from the cytosol of the cell. So under high ammonium concentrations, yeast cells tend to also exude quite a bit of amino acids, and that exclusion of amino acids is enhanced when potassium concentrations are low around yeast cells. So if you actually take away the 2 high affinity or the high affinity and low affinity potassium transporters in yeast called TRUC1 and TRUC2, you get a yeast cell system that is very sensitive to ammonium. And this is some data that was collected back in the early 90s when this yeast strain CY162 was developed, where they found that this strain was quite sensitive to high concentrations of ammonia, which grow the cells in only when you actually pull back the potassium concentrations to below 7 millimolar potassium. So on a typical CY162 cell line, if you grow at a normal ammonium concentrations in a yeast media at low potassium, they won't grow well at all. So using that system, we've been able to actually test how these AMFs might do in the context of ammonium toxicity. And so we found that when you overexpressed AMF2 in the strain, it allowed that strain to grow onto close to 150 millimolar ammonium. This is an example of 100 millimolar ammonium. So we're able to actually mitigate that potassium deficiency and ammonium sensitivity. And we've cloned AMFs from maize, from rice, and from soybean, and we show this very similar sort of phenotype where those select AMF2 type proteins can actually facilitate management of ammonium toxicity in yeast. We think that AMF2 is there to enhance ammonium uptake to the vacuole. We know this protein is localized in the vacuole and working as a proton antiporter. The thermodynamics of that would be basically protons coming out of the vacuole with the exchange for ammonium coming out of the cytosol and into the vacuole to relieve any sort of ammonium toxicity that might be associated with vacuolar or cytosolic ammonium accumulation. And we've also looked at AMF3, which is clearly on the plasma membrane and following typical chemical potential gradients across the plasma membrane. Protons are generally exported out across the plasma membrane through p-type ATPase activities, and that return concentration gradient would allow ammonium to be actually exported out of the cell in exchange for protons coming back to the cell. We've identified mutants for these 3 genes in Arabidopsis, and we spent a lot of time making sure these mutants are really clean and backcrossed. So if you remove the functional activities of these 3 genes, you get some interesting phenotypes. So collectively, on low ammonium concentrations at low or high potassium concentrations, the only one mutant combination that showed a phenotype was the double knockout of AMF2 and AMF3. So plants grow really poorly at low potassium, but can be recovered at high potassium on 2 millimolar ammonium. And you can then play with this a bit, because actually as you titrate more nitrate to ammonium, you can actually recover that mutation. So this is AMF2, AMF3, and AMF2, AMF3 double crosses. And as you titrate the more, add more nitrate to the relative amounts of ammonium, you start to get a response and you start to get growth, basically recovering as nitrate outcompetes ammonium. And if you look at the expression patterns of these AMFs, AMF2, as you add more nitrate, as you add more ammonium relative to nitrate, its expression goes up in response to increased ammonium concentration. And you see a similar response, but not as dramatic with AMF3. So you can take these mutant Arabidopsis plants and transform them with using to try to phenocopy the deficiency of the AMF2, AMF3 background. And then when we express the tonoplast AMF2 in the double mutant, you can actually recover the sensitivity to ammonium. Or you can overexpress the AMF3, which is on the plasma membrane, which will be pumping ammonium out of the cytosol into the apoplast. And you can also recover sensitivity to ammonium as you when ammonium to nitrate ratios are relatively high. One of the interesting things about this gene, or these genes, is that they tend to show their presence as plants start to senesce. So this is a before and after bolting phase in Arabidopsis. And what you see is a drop in nitrogen content, particularly in the older whorls of Arabidopsis, shown by these yellowing leaves after bolting. And if you look at that in more detail under high or low nitrogen levels, you start to see some significant impacts on seed development in Arabidopsis. So even on the single mutants, AMF2, AMF3, and the double mutants, you start to see a reduction in yield per plant, reduction in seed length in Arabidopsis. It's a challenge thing to measure, of course, for small seeds. Seed area, and particularly if you use a pulse chase N15 feeding experiment, you start to see a real dramatic drop in partitioning of N15 to developing seeds. So to summarize, I'm thinking on time, is that these are 3 proteins that operate in Arabidopsis. AMF1 is endoplasmic reticulum localized, AMF2 is tonoplast, and AMF3 is plasma membrane. And I don't have time to present other data, but we have comparative work in rice as well as in maize, showing very similar phenotypes. These things are expressed in response to ammonium, and they also respond to tissue age. And a loss of activity will really disrupt development, increases the ammonium sensitivity of these plants, and it progresses early senescence in leaves in Arabidopsis in rice. And it also will disrupt yield and redistribution of nitrogen into seeds. So these proteins have to be on to actually get nitrogen remobilized from the leaves and delivered into the seeds. So our suggested mechanisms at this point in time is that AMF2 retains ammonium in the vacuole, very important part of plant cell development to make sure it doesn't have high concentrations of ammonia, and it manages ammonium homeostasis in living cells. And AMF3 is activated basically to remove ammonium from the cytosol into the apoplast, and that's really important as those plant cells start to senesce and to liberate a lot of that ammonium back into the apoplastic space and to deliver into developing meristems and developing seeds. And I'll stop there. Thanks.
Thank you. Further time. Do we have any questions, Brian? Yeah.
Thank you, Grant. So you probably know what I'm going to ask you. What is the mechanism
What the role is. So I think these classes of proteins are essential homeostatic ammonium management systems in all plant cells. So they're lowly expressed. They're not that active, but if you disrupt their ability to manage ammonium, which is the constant flux of nitrogen that's flowing through a cell, you get problems. And importantly, I think they're very important in how you can then reprocess that nitrogen as a vacuole breaks apart. All that stored ammonium that's coming into a vacuole all throughout that life of that cell. If we're not officially remobilizing that, we're not going to be utilizing. So I think they just have a role in making sure that there is good flow of nitrogen through the cell system and can deliver it to developing tissues. The symbiotic data that I did present today is really interesting because if you turn these things off, the whole mycorrhizal symbiosis stops effectively, and they're present on the vascular membrane or the perivascular membrane, and they're really to mop up ammonium that's coming out of these fungal-based systems, and I think playing a very similar role in the nodule as well, which is subject to really high concentrations of ammonium that tends not to be seen, and you need systems there to manage that ammonium. So I think that's where their role is, and how we'll sort of play with them going forward to understand their functionality. But the interesting thing with rice, for example, there's only one of these genes in rice, so it's not a highly conserved rice. But if you turn it off in rice, you don't get seeds that develop well and may have a problem.
Yeah. What exactly ammonia is doing in the mitosis as we show that showing a decrease concentration.
What's ammonia doing in the seed?
Just because.
Well, I'm not sure if it's ammonia being partitioned to the seed. I think it's ammonia being released to be reassimilated into an amino acid that's probably being delivered to the seeds.
Is it assimilated there or is it being released from?
I don't know where it's being assimilated. We haven't done those sort of detailed analysis, but I would imagine that most of that ammonia either is being reassimilated by GS in the cytoplasm as those cells are being aged and releasing ammonium, or being reimmobilized or reassimilated in newer cells as that ammonium is flowing to those developing cells.
Or is it possibly storage proteins?
Is what?
Is it possibly converted into storage proteins? Storage proteins.
Yeah, well, that's a downstream after assimilation to an amino acid. So yeah.
Thanks. One more discussion.
Sure.
Thank you.
You're welcome.
Well, I'm going to have to introduce Sabrina. You may have seen her face on the times which she's actually been one of the very essential members of the team in terms of putting so first of all, thank you very much for all of your efforts and the success of the conference. Look forward to your presentation. Next piece of work for you. Real responsive transcriptomic comparisons of drugs in drugs.
So my today's topic for presentation is urea responsive transcriptome from comparison of rice genotype, contrasting for nitrogen use efficiency. Before going into the depth of introduction, I will just jump into the methodology. So we have used 3 different doses of urea for 2 contrasting genotype that is earlier identified in our lab, Aditya and Vikramarya. After 21 days of treatment, we have harvested the tissue and we have given the tissues for, we have isolated RNA and we have given the RNA for the RNA-seq analysis. After that, we have done all this in silico analysis, and after that validation process is going on. So this is the correlation and scattered plot for the RNA-seq data. As we are seeing here, all the replicates are correlated to each other, and the MA plot showing the counts data. So these are also given. Next, we have identified the urea responsive differentially expressed genes. So this is basically the volcano plot. Here, red dots are showing upregulated DGs, blues are showing downregulated DGs, and black are not up and down regulated DGs. Basically, we have used three parallel pipeline to just find out DGs which are differentially expressed in the contrasting genotype. Generally, people use only one tools for identifying the DGs, but we want to cover all maximum possible DGs. So here, two of my colleague and me, all of three use parallel pipelines, that is HiSet, Star, and Salomon for alignment. So after alignment, we have got a different number of DGs by all the three tools given in the pictures of each. So for further analysis, what we have done, we have just done the Venn selection. We have found the common DGs among all the tools, and we have included the common plus exclusive DGs. That is total overall DGs for further all the downstream analysis. Now, this is the geo-based functional annotation of all the DGs among different treatment. I'm not going to depth for every detail, so this is given on the slide as there is very restricted time. So these are the different categories among different treatment for geo annotations. Now, the main purpose of our work is to just, because there's so many genes, thousands of genes are available in the literature and databases for nitrogen use efficiency. They are telling that they are nitrogen use efficient gene, but we have developed in 2021 a paper on the development of a novel pipeline for short listing of important candidate genes for NUE. So we have, following that methodology, we have searched for the yield gene in the literature and databases, and we have found 6,588 genes which are yield related. Now we have done our Venn selection to find the common and responsive yield related genes, because in nitrogen use efficiency, two biological factors are there. First is nitrogen response, and second one is yield. So we have captured all the area because unresponsive transcriptome data we have got from our analysis, and we have searched the yield gene. So doing Venn selection, we have got the unresponsive yield-related genes, which are NUE genes. We call them NUE genes. So we have got 1,814 NUE genes for Aditya and 1,365 NUE genes for Vikramarya. So after getting these genes, we have done all the analysis, in silico analysis like geoanalysis, transporter, transcription factors. All the analysis have been done in these NUE-related genes, and we have found different categories of transporter and transcription factor in each of the families. Among them, we have found so many families of the transcription factor that are not reported earlier, that are novel. Even literatures are not available for that transcription factor or transporter. So we will explore them through wet lab validation. Next one is this is the method. This is the paper that I have given in the right side. This is our paper from the lab. This is a novel methodology which has not been earlier published for any crop. So we have given this pipeline, and we have used this pipeline in our this analysis also. So as you have seen here that we have found the NUE responsive genes from the Venn selection. After that, we have applied several criteria for high radical shortlisting, not single step shortlisting. We have applied multiple things. Like first, we have co-localized the NUE genes to the QTLs. We have taken major QTLs, and we have physically locate them, our DGs, to the QTL regions. After that, yeah. Okay, sure. So after that, co-localizing genes to the QTLs, we have just find that chromosome 1, 3, 5, and 9 are having most of the DGs. Earlier, we have also found that these chromosomes have most NUE genes. So these are hotspot for NUE genes. After that, we have found the common genes among both the genotype. These are 89 common genes. These are very important genes. We will see the regulation of these genes among the contrasting genotype, and this might be the most suitable and most important NUE genes. So these are the conclusion and future prospect. All the transcriptome analysis show the genotype specific expression of all the genes, DGs. Most important genes have been present in 135 chromosome. So these are the most important hotspot. These genes which are identified here can be used further for the CRISPR transgenics, and we are also doing SNP analysis. We have found SNP, but right now filtering is on process, so I have not given result here. But surely SNP will be used for Cas marker development, and this is a high throughput marker which will be used for high throughput genotyping through mass. Thank you so much.
Thank you. Very comprehensive.
Thank you so much.
So, yes, one very quick question. I'm not a molecular biologist or geneticist, but I wonder what kind of phenotype you're using.
Actually, we are using Aditya and Vikramarya. These are the two genotypes that have been shortlisted in our lab by my colleague using different criteria like germination-based criteria. We have shortlisted 21 genotype among the contrasting genotype, early and late genotype. Like early genotype are low NUE genotype, and late germinating genotype are high NUE genotype. So on that basis, I have selected this contrasting genotype. What? Sorry?
What is your definition of NUE?
Oh, NUE. Okay, this is the nitrogen used per unit, yield per unit nitrogen intake.
Okay.
Yeah, uptake.
And they are same harvest index or different harvest index?
Harvest index? That may be differ.
The reason I'm asking is because harvest index is directly linked to nitrogen utilization.
Yeah.
And so you should be looking at this differences within genotypes with similar or same harvest index.
Yeah.
And not different harvest.
Yeah, sure. Thank you, sir. Thank you for your suggestion. Thank you.
Again, sounds like more discussion. Okay, thank you very much for your presentation.
Thank you, sir.
And our last speaker, I'd like to invite Judah Ollie Kenny. Thank you. The title of the presentation is Progress in the Improvement of a Quality Protein Maize for Tolerance to Low Soil. Thank you.
Good afternoon, everyone. I'm Moloyede Kudros Tabiyo from Nigeria. I'm a plant breeder. I'm here to present the progress in our breeding efforts in improving the quality protein maize for tolerance to low soil nitrogen. Nitrogen is a very important element in maize growth and yield, but soils of most of the maize producing ecologies in sub-Saharan Africa, including Nigeria, are relatively low in nitrogen. Many low-end tolerant maize have been developed, but none of them is a quality protein maize, and that is why we are trying to develop quality protein maize with tolerance to low soil air to enhance its use. So the objective of this project is to see the progress in 2 cycles of recurrent selection and the improvement of the quality protein maize. In this project, we use recurrent selection. Recurrent selection is actually a cyclic method of concentrating gene for resistance in a plant population, and it has 3 stages. First is to generate progenies to evaluate. We take them to the field to evaluate in replicated trials. Then we select the best during the evaluation and we recombine them to form another base population. That completes the cycle, and the cycle continues. So here we generated S1 progenies from the base population, and we evaluated them under low N at the rate of 30 kgl per hectare using urea and high N using 90 kgl per hectare using urea. The field use was the field had been depleted of the soil nitrogen over years. So here are the details of the data compared. Our results. From the results, we have been able to complete 2 cycles in this projects, we realize that for plant aspects, which is the overall appeal of the plants, we rated that on a scale of 1 to 9, 1 representing excellence and 9 representing poor. So in the result, we realize that the overall appeal of the plants has actually improved from 4.3, from 4.8, I think, yes, in the initial cycle to 3.78 in the second cycle. We should reduce. Then for staggering ability, that is a sign of under low end. That has also improved. We also use scale of 1 to 9, 1 for excellence and 9 for poor. Staggering ability also improved from 4.68 to 3.6 in the second cycle. Antecedent seeking interval, which is the gap between days to 50% silking and days to 50% pollen shed, is also a sign under low soil low end condition. It also reduced from 5 days to 4 days in the second cycle. Grain yield also improved from 0.76 tons per hectare to 2.19 tons per hectare in the second cycle. And if you look at the heritability, it ranged between moderate to high for most of the data collected, except for antecedent seeking interval. And looking at our delta G, that is our expected gain from selection, we also see that it increased in the second cycle. From this result, we were able to make 3 conclusions. One, that there's high variability in the quality protein-based population for tolerance to low soil nitrogen. And secondly, we're able to detect that our selection have actually been effective. And thirdly, it is actually an indication that we'll be able to make better progress in advanced generation in the improvement of the quality protein mix. These are the pictures taken under a low soil condition on the field. The maize cobs with scattered grains under low and our quality protein maize kernels and our maize showing sign of plant death. That's poor staggering ability under low soil condition. I want to appreciate my Institute of Agricultural Research and Training, Nigeria, and IITA Nigeria for funding this project. I thank you all for your attention.
So are there any questions? So one quick question from me. So this is very promising in terms of improving issues. But there are also, I guess, repressed yields associated with. You make some comparisons of the expected benefits that you would get under these low nitrogen, the type of yield increases from the selection of.
Thank you, sir. Like I said, we have only completed 2 cycles in this presentation. The breeding project still continues. And from here, we could see that the yield have actually increased. And from here, if you look at the genetic gain, this is in cycle 1, it's actually increased. This is it. So we have, in the first cycle, we had 71%. The genetic gain was expressed in the percentage of mean. So we had 71%. And in the second cycle, we had 27. That means there's actually an increase, although it's a bit small, but there was an increase and it is expected that in the advanced generation, we'll be able to get something much better. Thank you.
No further questions? Thank you very much. I appreciate it.
Thank you, sir.
You asked all of the speakers, the session, please to come up front because there will be the presentation ceremony
I really appreciate the co-chair, Dr. Cameron. To facilitate him, I would request Dr. Mashita to join us on stage. Please come on the stage. Thank you all the speakers to joining us on the stage. Yeah.
David Cantor. We have an international literature workshop hosted at our university office this summer, and I have an announcement to make. If
No, that's why she just double wrong. So what is that?
Yeah. She's.
Can you
Hi.
hear? Perfect.
Yes, can you hear me?
Perfect. Awesome.
Great.
Okay,
Thank
so
you.
we're still preparing things here so we can let you know when we're ready. But works perfectly. And good to see you.
Okay. Good to see you too.
And
Thank you. Say hello.
hello.
Can you hear us?
Yeah.
Hello. Yes, I can.
Fantastic.
Great.
प्रेजेंटेशन वहाँ पे या बस पहली स्लाइड में एक स्पेलिंग मिस्टेक है members E है डाल दिया है
Ready? What do you think? Could I take 1 second to announce?
Yes, just right
You can
now.
do it right now. Yeah, why not?
Okay. So your presentation is this one, right?
Yes. That's great. Let's just take it.
Yeah, I'm just
Screen.
sharing on that. It's very.
So I'm gonna make it with an item.
You just come over here.
Yes.
Just
Good afternoon, ladies and gentlemen. I have a short announcement. It's a privilege for us, Aarhus University, Denmark, that we are the organizers of the International Nitrogen Workshop this summer. And there is a web page here. And there is also a deadline for abstract submissions. It has just been extended until March 15. So I would actually say that for all the audience here at the conference, it would be very nice to see you in Aarhus this summer. With this conference, we also each year organize a summer university on integrated nitrogen management. It's mainly for master students, but it could also be bachelor student English speaking even PhD students. We do it together with Hokkaido University in Japan. So it would be a good warm-up for the next conference in Kyoto. And this is also the same week of June, so as an N workshop. So feel welcome to have a look at the webpage. Here's also the sessions of the nitrogen workshop. You can have a look. Thank you for your time and attention.
I'm presenting.
Unity?
Yes.
For Unity?
No, this is the one about Dali.
Policy or the Unity?
Just the one slide, and then we'll go to the policy one after.
Okay. This one.
No.
Another. I'll just,
So if you go to the policy one,
so the ones I.
Okay.
and then you just go to that one.
Yeah.
And then we'll go back to the ones. This everyone.
Hello, everybody. I think we're going to get going. So the first thing that we're going to just say is that this session is actually in memory of a colleague, our dear friend Dali, Dr. Dali Nayak, who was working at the University of Aberdeen. She sadly passed away on Saturday. And certainly in my presentation that I gave, well, should have given earlier, one of the papers that I was referencing, she was one of the co-authors on. She was very important to this project, and her energy and enthusiasm will certainly be remembered. For those of you who knew Dali and would like to raise a glass in her honor, we'll be meeting at 9:15 by the palm trees at the hotel where the dinner is at this evening, if anyone wanted to join us. Thank you very much. Okay, so I would also like to share some good news. We are going to be joining the next two sessions in one. Woohoo! And no, that doesn't mean that you get to sit for twice as long. We're actually going to be making the first session slightly longer and cancelling the second session. Okay, smiles, please. Yay, round of applause, woohoo. Okay, before I introduce our first guest, who are the second session, but on first, I wanted you to be aware of this because we'll be using it in the policy session. So we're making it interactive. If you use your phone, hopefully either you've got 4G or Wi-Fi is working, point it at the screen and it'll come up with WooClap, and then we'll talk about the questions later, but this is just to set you up. Okay, so we'll now do the second session first. The reason we've changed the order of this is because it's great to have a proper introduction to what's happening at an international level on nitrogen policy. So we have two people who are coming up to speak. Oh, you're both at the table. They're both from UNEP. So, and I'm going to get your names wrong. I can just see this. Here we go. No, do you want to just change the slide? So it's Ningling will be speaking first, and then Mihai. Thank you very much. Round of applause.
Okay, thank you very much. So on the 26th of February, the 6th union will be held in Nairobi. So we expect 6,000 people will gather in Nairobi to discuss about the environment issue. So it's a very important forum. But for the nitrogen issue, we are very proud that Union 4 already have one resolution on nitrogen. And again, on the Union 5, the other resolution on nitrogen. So this is a very important decision on the nutrients work. On the 30th of August 2022, the executive director of UNEP invited the member states to nominate focal points and the co-chairs to the working group. And then the appointment of two co-chairs. One is Ms. Pooja from India, the other is Mr. Mihai, and also is sitting here in the panelist. So to be the co-chair for the working group on nitrogen. As of 10th of January, 87 countries nominate focal points to the working group. And 5 meetings have already been organized. The progress is mainly on the development of national action plan. A baseline screening of existing actions and action plans was conducted towards the preparation of a voluntary national action plan, and the findings and the recommendations were processed for being included a voluntary national action plan template with 10 action areas, which was further consulted within the working group. Those 10 action points include leadership and coordination, a monitoring network, guidelines, action programs, norms on wastewater treatment and discharge, emission norms on combustion, awareness campaigns, capacity building, public investment, and scientific technology advancement on reducing nitrogen losses. So the member states have started to request the UNEP to support in the development of national action plans for sustainable nitrogen management. And the Global Partnership on Nitrogen Management has provided technical support to the working group through information sharing, informal expert meetings, and direct support to member states. And the facilitating improved coordination of policies across the global nitrogen cycles include that it was agreed to establish an ad hoc task team to examine, among others, the possible TOR for an interconvention nitrogen coordination mechanisms. Compilation of substantive comments received from focal points after the second meeting of UNEP working group on nitrogen on the task team report and a template for baseline screening on nitrogen actions prepared. All the interventions from the focal points during the meetings of the working group have been recorded in its meeting report. The working group considered several options and the modalities for improved coordination of policies. Options for better management of global nitrogen cycle were explored in close collaboration with relevant UN entities. The Environmental Management Group held a Nexus dialogue on sustainable nitrogen management in April 2023. UNEP is mapping out mandate across the activities of UN organizations on sustainable nitrogen management. And UNEP is developing guidance on legal policy framework for an integrated approach to reduce nitrogen footprint from sectors. The current governance and the potential options identified for consideration by member states in the upcoming Union 6 include current governance strategy and approaches are fragmented and do not adequately address the global issues of nitrogen pollution. And the working group identified the need for coordination of activities and action and multiple agreements, including the Kunming-Montreal Protocol, biodiversity framework, and the global framework on chemicals, and invited UNEP guide how to address fragmentation of policies on nitrogen management. Collaboration between UNEP and the sector-specific international UN entities to increase access by countries to tailored expertise. Need to further support and guidance related to technical and policy preparation and implementation. The working group is currently the most important forum for member states to work together on sustainable management on nutrients with a focus on nitrogen. Still, for actions to be set in place, more time is needed to allow for further progress to be made. Going forward, additional national action plan areas may include monitoring, evaluation, and reporting. It is also recommended that additional guidance to be developed on preparing a national action plan. A harmonized monitoring framework for nitrogen could be developed, including procedures and indicators. The working group considered various barriers to sustainable nitrogen management, including challenges related to administrative and financial resources and the need for capacity building and technology transfer in developing countries. Looking ahead, it is also important to recall that many countries have not initiated work on national action plan. Consideration could be given to knowledge management platform to facilitate the sharing of experience and best practice, recognizing the value of the GPNM has provided to date and called on GPNM to continue providing further support. Further work should be undertaken to address unresolved matters such as terms and definitions and increase a common understanding of the issues related to nitrogen cycles and possible other nutrients and their connections with biodiversity, climate change, and pollution, as well as approaches to address them. In the meeting, many experts mentioned about the global partnership of nutrient management. So this GPNM played a scientific support role to the working group. And so we wish that more partners will join this partnership to share information expertise and also advance scientific research. So this is the overview of the progress of the UNEP working group on nitrogen and also the brief introduction of GPNM. We wish that in the upcoming Union 6, there would be a new resolution on nitrogen, but we will keep you posted. Thank you very much for your attention.
Thank you very much. There were certainly some points there that I definitely agreed with, such as making sure that the administrative issues within the ministries is actually dealt with. Trying to get different ministries to talk to each other can be very difficult. I'd like to call on Mihai as well from the UNEP, who's going to do a double act, both now and then in a few moments in the panel. So on you go.
Which one it is? This MISCI? Should be this one.
Thank you. Good afternoon to all. I'm Mihai Constantinescu. I'm co-chair at the working group, UNEP working group, but I'm from Romania and I'm director in my country coordinating management unit for investments in sustainable nitrogen management. So I took this task at the end of 2022. As you presented, we are now 87 countries discussing about nitrogen and trying to put into place the provisions of the resolutions, and actually the terms of the references of our group are based on the provisions of the regulations. So we are considering this number not to be quite sufficient to have the critical weight for advancing, and at the level of UNEP and at the level of the Committee of Permanent Representatives, we are pushing for more countries to designate focal points. Officially designation is important for us, and we're hoping that we'll meet 120 countries by, let's say, in the first quarter of this year. I'll go through achievements and let's say the gaps that we still having on the progress towards the implementation of the resolutions. One by one, as mentioned on the left side, you'll see the terms of reference provisions as extracted from the resolutions. And on yellow, I just put a bit what remains to be done, considering that we do have a secretariat from the UNEP, and this means we do have the UNEP commitment of going ahead. Only from the end of 2022, and the second meeting of the working group was in January last year. We've been asked to prepare a roadmap for further implementation of the resolutions. We've done something. I would say this was partially done. I have the roadmap presented here, but this refers actually to the work of the working group up to the UNEA 6. So the meeting of the United Nations Environmental Assembly. I believe we should look further, and this remains to be done in the future. Of course, we follow up the task of the resolutions. This was actually a roadmap for the working group that we drafted at the beginning of April before the meeting in Bucharest, before the third meeting of the working group. And we pretty much stick to the proposed plan. As you may see, the core of our actions were around an action plan template ready to be used by the member states, options for improved coordination of policies at international level, and exchange of experience. And there is a certain time sequence that we need to follow, and we still need to address in the future. We've been asked as a working group provide technical and policy recommendations, options to accelerate actions to significantly reduce nitrogen waste globally by 2030 and beyond. This type of wording actually was, as I was told, negotiated very hard before the UNEF 5.2 resolution, because it's supposed to have here, or initially had here, the global aim of halving the nitrogen waste. But after the member states negotiation, this is what we all end up with. Well, I have to say that the template for the voluntary action, national action plans is quite comprehensive, but it does missing some points that we'll need to address in the working group. It doesn't have a time frame. It doesn't have yet and monitoring and evaluation system. So we will need in the future to address this, but actually we want to have a step-by-step approach to involve the countries, to focus at the beginning on the actions, to see the reasoning of the actions, and to motivating them being part of the working group. And now remains to follow the resolution provisions and to make progress on completing the National Action Plan framework that we developed. Share information on voluntary national action plans. This is what we done even from the beginning. Mr. Ning Liu from UNEP presented that at the beginning we started with a screening baseline document, collecting information from the member states and try to see what was implemented and what was not implemented. I'll make some comments on this further on. Identify possible modalities for the options being considered on coordination of policies around the world. Well, at this point, we all agree that there is no option to select or a single silver bullet. We work very much and closely with working group, and I do believe that this is the main factor that is intensifying the discussion and progress towards implementing action at national level. GPNM, we found a partnership in GPNM that in the next period will be revitalized through new members in the steering committee and new members as well as part of the GPNM. Nexus Dialogues, it was already a meeting and we'll continue on that. I were here. TFRN, Task Force on Reactive Nitrogen, which is doing quite a good work, and I've been part of their assisting their work in Germany last year. FAO, UNICEF, research projects, and so on. So it should be like a set of type of intervention, and I miss here to include conventions and protocols, which is quite a challenge. Provide a forum for dissemination and communication of information to related to UNEA 4.14 and 5.2. We done this, of course, through the working group and GPNM, and it's still to be done. A lot to be done on this. Participate in experts group meeting roundtables. This is done again. Well, before going ahead to a different perspective of our work, I have to say that at this moment there are like 15 member states that recently ask and officially ask support for the UNEP to initiate actions in their countries and to actually to put in place national action plan. This involves setting up a committee between different ministries, relevant ministries, with Minister of Environment and Minister of Agriculture and Minister of Transportation or Minister of Energy, depending on the priorities and the specificities of the countries. Of course, involving the proper stakeholders is a challenge, and this was underlined as well for the representatives of the countries that ask support. And it remains with UNEP and to find the ways, and will help as well to find the ways and to find the financial means to support and address the request. It means something because we also realize that most of the countries, even after initiating the analysis of the baseline screening documents, most of the countries are having a few actions or zero actions implemented. And it's clear that nitrogen is still having a very low visibility at the level of most of the UN member states. Somehow at the end, we try to look differently to see what we missed from our discussion, the level of the working group. And we made the screening as well through the publications. Maybe we're supposed to, and this is something that we'll address as well in the future, to link a bit our topic, the nitrogen, a bit with the food security. And we all know what's happening now in Europe with street demonstrations and with the farmers. Fertilizers is a topic as well, very much indicated by quite a lot of developing countries, and they will need regulations for this. And as well with innovation. Looking as well in a timeline how the scientific publications, what topics were addressed in the scientific publications. we see that agricultural waste, biofertilizers, plant growth, methane as well, which is linked with nitrogen, ammonia, and agricultural technology. It's something that is coming in the recent studies that were published. And we are also having GPNM to make the link between different stakeholders and to try to create more debates on these topics. Thank you very much.
Thank you, Mihai. So does anyone have any points or questions that they want to raise from the audience? No? Okay, well, let's go on then. If you could, just while the slides are getting put on, I realized I didn't actually introduce myself. I just assumed everyone would know who I was because I'm normally so loud. My name is Keisha Avis. I work for Ricardo and I've been part of the South Asia Nitrogen Hub, working with Annie and Roger on stakeholder engagement. Okay, so for those of you who weren't here when we started, we're going to be making this interactive, so please look at me. And we're going to be using yes. We're going to be using something called WooClap, which is a way of asking you questions that you can answer online. So if you have your mobile phone and you're either connected to Wi-Fi or to 4G, we're going to be showing you a what are they called? QR code, that's the word. They're going to be if you can have a look at the QR code, and then that is going to take you to WooClap. You then need to enter the event code in the top banner, and the event code is there, FNAONB. Okay, and then let's see if we can test it. Right? Has everyone been able to access WooClap? Anyone have an issue? Okay, that was your first test of interactivity, and most of you failed. So you've got a better try next time. Okay, so what is the first word you would associate with nitrogen policy? We have 0 answers so far. Subsidies, very good. Implementation. Oh, I like that. People actually have implemented nitrogen actions. That's great. Weak. Yeah. Delay. Wicked problems. Yes, definitely a wicked, complex issue. Agriculture. Yes, but hopefully after this conference, we all realize it's not just agriculture. What else? Urgent. Yes, urgent is a very good one. Annie really wants to be in control. Nitrogen waste, yes. That is two words, by the way, just in case anyone was wondering. Fertilizer, yes, but also interestingly, a lot of it is actually to do with, in agriculture anyway, manure and livestock waste, but we keep coming back to fertilizer, maybe because it's easier to talk about. Necessary, nice. Integrated. Environment, yes, and it's multiple. It's air, it's water, it's health. It's our natural environment, it's our urban environment. Right, regional differences. Again, that's 2 words, but very relevant, so I'll allow it. Nitrate, yes, okay, we're getting into the different species. Credit system. Yes, I suppose the way that these policies so far are set up in some countries is definitely enabling people to buy fertilizer with things like credit systems, but perhaps you're also talking about another way to enable people to get more fertilizer. Okay, this is making me dizzy now. Right, okay, does everyone know how to use WooClap? Yay, well done, Bagatti. Okay, that is a prime example of proper audience participation, okay? And I didn't prime her up, I promise. Right, let's go to our, now I'd like to introduce Professor Roger. Do you go by Professor Roger? Who is going to introduce the session. On you go.
Thank you. Keep it up, that's fine for the moment. Okay, thank you, Kesha, and that's great. So I'd like to know how many people want to be using WooClap but didn't get on in time. I couldn't because I was facing the wrong way and everything else. Do we need to do that again? If you don't speak, we will just carry on. Okay, that's great. So no excuse if we don't get your answer. So I'm Roger Jeffrey. I've been a partner in the South Asia Nitrogen Hub over the last 5 years. And one of the things we've learned in this hub is the importance of bringing scientists into policy dialogues, various kinds and in various contexts. We took a while to get round to doing that, and we had to overcome a fair amount of uncertainty on the part of some of our natural science colleagues. What we wanted to do in this session is, and I think I've got the right way of presenting this, but of course it may not work, is we want to encourage scientists to be involved in engagement with the policy process. So the first thing is engagement. It's encouraged you to engage. And the way you do that is various. You can think about different ways, but we hope this session will give you some encouragement to do that. The second thing, and one way of doing that is to give you some options, to suggest some ways in which you might do this. And the third one is to say, think about the partners that you might get, or you might find to invite you, or you might want to invite to join you. The key thing that I think we want you to understand is that when we've got scientists into this process of trying to help bridge the gap between science and policy, they've loved it. They've enjoyed it. Not everybody all the time, but they overcome a certain amount of uncertainty about getting involved, and they come away and say, well, that was one of the best things I've ever done, or I realize now that it's not so difficult. So that's the kind of takeaway messages we want you to have at the end of this session. Yes, it's possible. Yeah, I can see ways in which we can do that. And yeah, I can also see who I can use, who I can be with when I do this, who will lead, and so on. So that's all I'm going to say in terms of now of what we want the session to achieve. The second thing, as you've already noticed, is we want to make this interactive. It's 5:30. It's the second day of a conference. You've been through a lot today already. We're going to finish at 6:15, full stop. If we haven't finished the discussion, we'll just call an end so that you can go off and get back to the hotel and have a drink or whatever else you want to do. So use the time as effectively as you can for what you think you might get out of this. We will have presentations and then an opportunity for you to interact, and then another presentation. So there'll be a series of gaps. There won't just be people like me standing up the front and talking to you and trying to make sure you stay awake. Well, obviously I've failed. Okay, so the first presentation will be from Mihai again, who's going to talk about not what UNEP is doing, but what he thinks we could be doing. Okay, you're going to do it there. Yeah, yeah.
So for me, again, it's my time to put into the discussion some key points about the relation between research and policy. First of all, I need the opportunity to say that we do have a working group at the national level because of the science results and because of the science community. So this is good. This is normal. It took a bit more time maybe than expected, but we do have at this point the UNEP commitment. We have a very strong secretariat and as well the technical persons since precisely. And I would say that the administration we not set up many evolution, it's step by step. Well, looking at what was done, we have to admit that at this point, we do have a geopolitical interest on the line. Still on the low utility, it's not like climate change, not pollution, it's not like biodiversity, but it's growth strength. And it's at that level that we can go far. And for this, there is a need to look at the constraints, at the barriers. And I would say that the scientific community is at the barrier. Where the barrier remains the awareness of the countries. And we do have developed countries, and we do have developed countries. And some barriers are in both sides. I could hear arguments that, well, my country done quite a lot, but there is some progress. And there are countries, quite many, I would say, that have done not too much. But it's good that there are signals, so there is an interest. And the working group does have all the strongest countries. Say, and it does as well have the interest of it's alive. Still, in case of accelerating the progress and the awareness of the this is the main question. It's something that I'm addressing to you as well to see what
hello everyone.
He's done. Do you want to keep playing now?
Yeah. Yes.
Hello everyone. I'm Gislaine Cunhazeri from Brazil. It's a privilege to participate in this.
Sorry, hang on a minute. Hang on a minute. I'm just trying to get your slide played automatically. Gislaine, just give me 2 seconds.
Awesome.
So I have a recorded slide and also a slide without any record. Then you may choose anyone, you can use it.
Well, I think go ahead. I managed to work it out.
Okay, no problem.
Gislaine, are you there?
Yes.
Okay, if you want to do your speak, please do.
May I share my screen for the slide?
No, just if you want to talk, because we have the slide already up. Is that okay?
Okay, great. So can I start right now?
Please do.
Okay, thank you. Hello, everyone. I'm Gislaine from Brazil. It's a pleasure to participate in this policy panel. I'm excited to contribute to our discussion today on nitrogen policies in Latin America. Nitrogen pollution is a significant environmental issue in Latin America, caused due to nitrogen emissions from productive activities to meet the increasing demand for food and energy. While the agricultural sector stands as the largest emitter of nitrogen, countries heavily rely on it for economic growth and overall development. Currently, most countries lack specific, exclusive, or integrated policies for nitrogen pollution. Despite some inclusion of nitrogen issues in existing measures, common obstacles hinder the development and implementation of more effective policies. Some of these challenges are: 1. Economic pressures to maximize agricultural productivity to meet market demands and global trade. 2. Poor governance, political instability, diverse economic conditions, and competing policy priorities all over the region. 3. Limited awareness and education regarding the benefits and drawbacks of nitrogen utilization. And 4. Lack of nitrogen researchers in agenda setting and policy-making processes. Addressing these and other obstacles requires an integrated approach involving collaboration among governments, research institutions, agriculture stakeholders, and environmental organizations to develop and implement effective policies that balance agricultural productivity with environmental sustainability. The differences in nitrogen policies approaches or the lack of dedicated policies among countries can profoundly impact regional and international agreements in many ways, including effects on agricultural productivity and trade dynamics both domestically and overseas, impacts on the global nitrogen cycle, and potential diplomatic challenges due to transboundary pollution, including issues in resources management, such as shared water bodies, disparities in strategies and priorities for addressing climate change mitigation and adaptation, and impacts on international relations and reputation, influencing diplomatic relations, trade partnerships, and global collaboration. Harmonizing regional and international agreements is critical for integrating environmental sustainability and economic factors into nitrogen policies, fostering effective global cooperation and shared objectives in nitrogen management. Finally, I want to emphasize some specific challenges in formulating effective nitrogen policies in Brazil within the agricultural sector. First, Brazil has extensive agricultural landscape with diverse climates, crops, and production systems. Represents a challenge in formulating policies to meet specific needs. Difficulties in fostering dialogue and coordinating efforts among diverse stakeholders, including farmers, governments, environmental organizations, and others. Political struggles, exemplified by the influential Parliamentary Agriculture Group, which constitutes more than 60% of Brazilian congressmen and consistently opposes environmental laws and initiatives in favor of agribusiness interests. And policy coherence issues arise between the promotion of sustainable agricultural practices and financial incentives in the ABC Plus plan and the recently launched National Fertilizer Plan's objective to produce fertilizers aiming to reduce import dependence and enhance Brazilian agribusiness globally. Two critical issues emerge while offering benefits to Brazil. Firstly, the fertilizer plan lacks specific measures to mitigate nitrogen pollution. Secondly, the potential availability of cheaper and more accessible fertilizer may discourage farmers from adopting sustainable practices in the ABC plan, posing a policy coherence challenge. In conclusion, addressing nitrogen policy challenges in Brazil requires a flexible strategy incorporating education and outreach. Ongoing evaluation, refinement, and collaboration involving stakeholders and nitrogen researchers are crucial for ensuring efficacy and sustainability in nitrogen management practice in the country. So that's all. Thank you for your attention. Hello, everyone. I am Gislaine Cunhazeri from Brazil. It's a privilege to participate in this policy.
Hello, everyone.
No, we don't want to go out again.
Hello, everyone.
No. Yes.
Okay, can you with that? Need to go back to the.
Yeah, you too.
Do you think?
No, I'll stand.
Okay, will you go?
No, you carry on there. Yeah. Which one?
Yes, this one. Yeah. Thank you.
As you can see, we're not exactly on top of this technology. So we're going to give you another question on the interactive model. But I think there are a couple of questions that I'd like to put to you to see if you can come back with any questions or comments to either of those first two presentations. And we'll ask the other people on the panel here also to respond if they can to what some of the things you can say. So one of the things I think that comes out of this is that awareness, that's something that we might talk about. Awareness in many countries is not present for nitrogen. So let's turn this around as an opportunity. What can be done? What can you do? How can you persuade people, suggest to people, encourage people, other people, yourself, what could be done? In your view, how can scientists contribute to awareness raising? Now, we're going to get another question as well, so if you don't want to stand up and ask a question, you'll get an opportunity to make some comments on the screen, right? You're going to get now a question that you can ask.
Well, yes.
I can't see what's going on. Okay, so these are the kinds of answers people are giving. Now it's an opportunity for you to disagree, to say, pick up on things you think are most important. Let's make this a little bit more interactive. Well, it's impossible in a building like this, with everybody spread out, they're about to see what's going on. I can't see hands going up.
Over there, Roger, over there. Is it?
Yes, over there, great.
There's a microphone coming to you.
I guess scientists have had quite a significant role in the climate change policy, in public debate around climate change policy, and perhaps a bit in the beginning. I guess, what are the lessons that us take from what's happened in the climate change arena that we can apply to this nitrogen challenge? The question is something that they discuss in the lobby before entering here. We did a panel, a science panel on nitrogen, and on this unit, most probably something before. So we all know that we have IMS project, getting quite a lot of good results, the patients on the way. And IMS gave a lot of support to the process. I don't see so much of it. I'm sure they will some establishments as the head of the IMS. But we are now at the moment when the process it does need some partners. And it's not only the climate change, process factors supported by the science. So this is discussion not only that we discuss now, it's just the past starting with the end of the last year. So it's something that will require a proper setup, and I think that vision is there.
Yeah, okay, so I was going to say something about this in some of the points I wanted to raise, but it's, yeah, I think we definitely can look to other policy domains or lessons, and certainly climate is more advanced in terms of, in terms of stakeholder engagement and thinking about gender issues and other kind of aspects, more of the social sciences, indicators, targets, so that, there's plenty of in that domain, as well as it's linked anyhow to
So, for the long-term protocol on the ozone depleting subsistence, that is one of the most successful environment agreement. When we look back at history, like in the 1970s, two scientists they found that the CFCs, chlorofluorocarbon, may destroy the ozone layer. And then in 1985, three scientists they found the ozone hole above the South Pole. That got the international attention. Then 2 years later, the International Convention on the protection of ozone layer was set up, and then became one of the most successful story. So the scientists play a very important role in the environmental protection, environmental issue. So most of the participants in this conference are scientists. So you are playing a very critical role. So your research, your funding, that can disseminate very good information to raise the awareness, to get the attention of the policy maker to do something. More questions or comments from the floor? Yeah, Adam, over there. Sorry, a long way down.
While the microphone's going, can I make a comment, Roger? I was just thinking that one of the things that might have to also learn is how to deal with fake news, because I mean, obviously, climate scientists might be respected now, but there was for a long time, they were told that they weren't right, and there are some policymakers who would certainly want to listen to them, or not listen to them, but and listen to others.
So, Roger, I wanted to respond to the sort of a personal action that I have to the question you said, what more can scientists do? Because I am no longer just a scientist, but I'm also a climate activist. And I have dedicated some of my time to local voluntary climate awareness issues. And whilst I don't suppose that's something that everybody in this room wants to do or has the time to do, I think it's something to be scared of. And there are huge numbers of community active groups who probably denied to have scientists give them talks and or even support their activities. And one important point of those climate action groups that are spreading up all over my country and probably everybody's country is the governments do kind of listen to them. So I imagine they have some benefit in policy.
Yeah, thanks, Adam. I think that's a great way of seeing this, that as scientists you can be a prompt, you can be a resource. You don't have to be the person who tries to go on television or leads the march or does the other things that activists do. You're there to give them the kind of information that's necessary and useful. There's another hand up over there. As Fisher says, these things are not straightforward. There will be people from the fertilizer business saying, "Oh, you need more fertilizer. Let's stop worrying about this." And scientific knowledge going into that debate is very important. You have it already. One of the things I think we found in the South Asian nitrogen hub was that people they didn't have their results yet. How could they talk? You don't, in the sense, you don't need your own research results are adding to a body of knowledge you know better than anybody else. So communicating that knowledge, the existing knowledge, is something that you can do and nobody else can.
Yeah, so to pick up on, because the IPCC has mentioned and Montreal Protocol as well, both of which obviously have scientific panels. And we've mentioned a few times during the week the various treaties. There are a lot of scientific panels out there that are associated with the treaties. So from the scientific assessment panel, they might not control N2O at the moment, but they just had their most recent term of reference for the next triennial assessment passed. That was in September last year. And that they've got a mandate to look at N2O on that. It's going to be at that. So there are lots of shit there. You know, people do want to get engaged on that kind of thing. And obviously we want people who are really in the know about the whole natural cycle because N2O there, so they can be given some due prominence and due attention. So looking for opportunities like those panels to really.
Okay, thanks very much. I think we'll move on now to the other two presentations we've got, and then another opportunity for questions and comments at the end of that.
Hello everyone. I'm Annie. Yeah, just to make points. Yeah, what we're talking about here is next steps in policy, actually policy research, where we go. So I kind of had three key recommendations. So basically multiple scales, active engagement, and the third was looking to other policy domains. So what I wanted to say about multiple scales is that, yeah, we need to be thinking about all these different levels. As David did say in his global study, actually tracking what's going on at the international level is fairly straightforward in terms of what policy is being developed or has been developed. But the gaps lie when we go beyond that, and that's the national to sort of local level. And so what many of us have been working on these kind of national assessments, this is something we've done in UKRI, SANH, one of our activities. So we did a collection for South Asia for the national level policies. What we haven't really done is a sub-national level. And it's just to reflect on that because in Pakistan, as one of the countries, provinces, provincial level is where really, the sort of key policies are coming out. So inevitably, we collect policies, from these provincial level, and also in India at the state level. So we collected 180 policies, and I'm sorry, I don't know if he's in the room, but went ahead and collected another 110 policies at state level. These are key when we're looking at what relationship these policies have with nitrogen management. And it's just to emphasise really that sub-national level, because at least that area deserves certainly more attention, because a lot of the cool things we see happening are at these local levels, as we heard this morning, what can happen in different communities, even with cities, or whether it's a state. So one really cool example is Sikkim, where it became 100% organic in 2016. So anyway, so multiple scales. And the second was this active engagement part. So obviously those involved in the society governance sessions are hearing more about participatory approaches, action research. That's where we're actually trying to, create impact, create change through the process of research. And that's why engaging and sharing knowledge or creating the spaces for different stakeholders to share knowledge. So not just a one-way, we're the scientists, here's the evidence, listen. But it's two-way, three-way, the sharing that happens there. So certainly, yeah, the active engagement. And then thirdly, it was just to comment on, learning these lessons from other policy domains. I mean, we obviously know there's a lot of policies in place that have a relationship with how nitrogen is managed, and that's how we, done these policy analyses. But we can look at what frameworks have already been in place. And so I was going to mention EU Common Agricultural Policy that I used to do a lot of work on, but I mean, they already had indicator systems, and there's ways to look at the baseline, the inputs, the outputs, results, impacts, and what the challenges were there. So, we can really sort of learn lessons and avoid mistakes. It's not just to say these are absolutely best practices, but within all of these different policies, you can see, we can learn lessons to learn, if you're going forward with national action plans, how can we sort of, look to other policy areas to learn lessons. I was also going to comment on RED Plus, so I've worked in forestry and reduction of emissions of deforestation and degradation. So some people call red plus failure, other people say it's still getting there. Anyway, the point was anyhow, there's still lessons we can draw from what's happened in this domain too, especially again, as I mentioned, with gender mainstreaming efforts, which are now, you know, and also the different phases of REC. So actually having a phase where you actually build capacity and build, you know, take the time to prepare people for when implementation actually comes. And you know, this is where you can address issues of equity, for example. So yeah, so I'll leave you these three recommendations.
So I have some few things I would like to highlight that are short. So one is connecting somehow with what Mihai and Ghislaine were pointing out. Or here I'm not talking about space, which I think is scary and complex, and we don't know how to play with. In many ways, it's a little issue for learning or like handling that multiple spatial scales or also temporal scales depending on what we're looking at, right? And when we think about governance of political discussions, it's more complex, right? So when I'm looking, when I see what you're saying, the work of that, you imagine working group, right? Highest level of discussions about the policy or where a human member says, probably. And then we have what we saw, which is saying, thinking about treatment. And I think the next here is that we need to build capacities in research. We need to build, you know, fill the gaps in some data associated with it, right? And at the end of the day, remember that even though we're working at these higher levels of international negotiations, we depend on nation states, we depend on at the subnational level on communities and science to produce knowledge that will inform higher classes. I think today that like awareness in here is something about either like awareness or a willingness to be part of political processes that we support, right? And that's something that not here. I think it's important to keep in mind when you think about the science and policy of how our work can contribute to those processes. We think about the scales of where we can practice, right? And what are our entry points and so one thing that I would like to highlight. The other thing which I think is very clear from this conference Spanish policy, that even though we're talking about policy, we're coming from different disciplines and backgrounds. And we are kind of very explicit about the need to build interdisciplinary networks and interdisciplinary the policy needs to be informed by what is happening in a biophysical dimension, what is happening with the flow, sort of emission for the right? But the other way around, you need the goal is to create policy-oriented wisdom, right? And especially when we're thinking about the stakeholder engagement, right? If we agree that the stakeholder engagement is really trying to pass policy processes at two levels, that means that we have to have a better understanding of how the stakeholder engagement needs to have and what that means, right? Probably needs to engage with people that have experience of training or feel more willing to, right, engage with communities outside the scientific discipline or even within it, right? So I think that's also very important. Something that I'm very, very interested and I would love to hear anyone who's doing work on this has appeared in several of the presentations during this couple of days. Is the question about social movements, not only as a way to engage with activism, right? All those movements are beneficial to us, right, or to the national agenda, but also social movements that can be appearing against, right, or move, right, against the growth, right? And that's something that is very important for natural science, the policy to pay attention to those areas of potential future conflict, right? right? So if anyone is doing some research, which is about following these farmers' protests, for instance, like this, which are very think about that. And I would like to finish with maybe something that Patient said about thinking about truth, right? And post-truth, right? Where we are today, right? And I think it's important to realize that, maybe connecting what I said earlier this morning, right? We are not in the world of 1970s, all right? We don't live in that world anymore where science and scientific knowledge has this kind of authority, right? That can create consensus in an easy way. It has never been easy. It has never been as before. And something that is a great theory. What we know how scientific change happens, how stable to implement policy is never that easy, right? That being said, I think we have to acknowledge that we live in a world that is very different, where how people perceive knowledge, how people perceive scientific expertise, right, is way more complex. And that means that probably we need to think in more complex ways about how we want to engage with the public and who is the public, right? When we talk about awareness, right? Awareness of what and for whom, right? We're talking about the public awareness of civil society in general, believe that nothing is a problem. Or we're talking about politicians willing to engage in political processes. So I think it's a problem that goes beyond awareness, right? And can be informed or disinformed depending on how we're engaging and providing knowledge in the process. And it doesn't sound promising, I know, but I think we have to be aware that we're not in that world 20 years ago or before that, and we have to come up with strategies that are more complex, pay more attention to different stakeholders and actors that are acting.
So now when we talk about policy, we often see that scientific evidence based policy. So the scientific research is extremely important. But the policy of countries they're very careful to make any policy regulation. So they need very strong scientific evidence. So then your research is very, very important. And also, yeah, the idea is communicated with the scientists. Sometimes I noticed that the publication is very important, but how to translate those scientific research results into policy somehow is not easy. We have to make the policy understood. Those kind of very technical things is also not easy. Also, the scientists have to pay attention to that part to translate your findings into policy. You put effort. Also, yesterday I noticed one professor from Nepal mentioned that female farmer may use more fertilizer. So those kind of research is very interesting. I think it also has the implication for policy that we need social scientists involved in the process. We also need the economists to give us more data. Then it is also very important for the policy making process. Okay. So I think this is an opportunity for anybody else who has something to add to what's been said already. I think we have another question. Let's see if we do. But there are one or two people in the hall who want to say something, which is great. So there's a hand up there. I think, well, this chance. One of the comments that came up was scientists are not policy people. Which I think is true. I think it's obviously true. But that's not the point. What we want to do is engage with people who have a role with policy. It may be with farmers, it may be with companies, it may be with junior civil servants, it may be with the local neighborhood watch association, the neighborhood residents association. doesn't have to be very often. It's a spreading of news about nitrogen. Every time we do that, somebody else listens or begins to understand what's going on. Yes.
Hi. So this is my observation about how natural scientists actually move away from having any idea about how policy is. And it starts right at grad school, where we are drilled into like the natural sciences in such a way that we have absolutely no idea by the end of grad school that what is really policy making about. So there is a disconnect right there. And it just continues from there. I think there has to be some, back end preparation for graduate students, right, at the grad school level about policy making. Yeah.
Yes, I think one of the things that we've learned more recently, and not only what you were saying about how science can affect policy, but also how science can be ignored in policy, but also how we don't all have the skills so everything might be necessary. So this is something that we can do just because we learned at graduate school. We can't learn everything. One of the things we miss in the South Asian Nitrogen Hub is that we didn't really have a design now. So yes, it's a great idea. Policy briefs, lost it now, but science work with NGOs, but certainly there was the need to simplify, produce simplified reports. We very often don't know how to do this in a way which is both simplified and accurate. So engaging with people who have that experience rather than saying we have to do it ourselves. I think that's the thing we should.
And just one more point, we have to what it means like to translate your work to policy. But actually in grad school, you're taught to write in such a way that nobody can understand your English. And that's a big problem. But what the natural scientists write their publications and the policymakers, they really are, and this is the biggest big problem of the other hand. If this is so, and then on the other hand, and this other hand problem has been plaguing natural scientists all the time. This and that, and at the end of it, the policymakers are, social scientists are not clear what they're trying to really say. So we have to improve the way we write our journal articles every.
I think, yes, every discipline is guilty of trying to make sure that nobody else can understand what they're saying. I'm certainly not going to say that sociologists write clear English. But of course, the other problem is that if science is held in English, there may not be local terms, it may not be easy to find ways of talking about the issue we're talking about in Hindi or in Farsi, or whatever it might be, whatever language is local. Many languages are local to work. So these are skills that are there if you look there, if you ask. And I think that's a big takeaway message to me. Once you stop thinking that it's your responsibility, it's something you work with other people to do, then things become a lot easier. I said we finished at 6:45. I think it is 6:45. I certainly don't think we should hang on in the interest of filling up more time. But if anybody does have a burning point, we could point in, right? No? Okay. Yes, one more.
You're talking to some people who are involved in science policy. Some of them have encouraged me and others as scientists that we should actually work directly with NGOs or more competent, perhaps, or more experienced, and actually directly engaging in policy to help do some translation. And I'd be curious what the panel thinks about that approach versus trying to more directly.
Oh, sorry, I kind of that education was asking. Yeah, okay, sorry. I'll try again. I'll say some questions. Yeah, and the role of NGOs has been quite interesting. Firstly, just to comment on that, it's just in terms of the context as well, where we say, you know, Acacia particularly, that's not for this actually, but she, you know, working with CSOs and their impact with policy, is particularly important in the case of Scotland. She worked for an NGO that was a great change in policymakers coming to them for information, whereas that situation is quite different. We're talking about the role of NGOs and CSOs more generally in South Asia. I mean, we've done that by stakeholder mapping, so you can see relative interest and influence through this kind of mapping approach that Cynthia talked about yesterday in the presentation. So yeah, the role of them differs, but certainly, yeah, that they are important, and having that as a gateway to also have sort of meaningful change. Because actually, a lot of us are probably quite skeptical about, even when we have good policy, do we see it well implemented? So yeah, and then what do we, you know, trying grassroots and bottom up and making that way there. So really, it's not it's policy, we still create change regardless. So I'm sure someone else has something else.
Can I add something else? So thinking about maybe connecting to the discussion previously about scale. So NGOs can operate at different scale, right? And we tend to think probably about having the needs is a big kind of NGO support campaign. But NGOs are also playing a very important role at a very hyper local level, right? I'm thinking about, I don't know, what local water pollution, right? We're working with communities today, which is very important, right? And having a scientist or an expert that can provide or help develop programs in science, so it's important NGO at that level, right? It's very, very important. It could bring impacts that will be way harder to achieve a larger scale, right? So I think there are multiple ways how that kind of work can happen, right? And I think it's important to pay attention where exactly those NGOs are working on, like what kind of topics, what kind of engagement they are producing those communities, and how our expertise is thinking about your work, would say, on biogeochemical processes or global, right? How do we translate that to target specific kind of policy frameworks at the global level? Right? It's a kind of work, like most of your work focuses on one region or one place, right? Subnational level. This is way more to do, and because we know that having a nice clinical issues that air pollution, water pollution, climate, those NGOs are already working on those issues. But we got to figure out what is our main point with those issues to be able to then probably create awareness on them, but not in some easy way to continue to go forward.
Can I just say, the SANH, just as an advertisement, will be producing fact sheets just on that point to show the interrelationships between nitrogen and biodiversity, both in mountainous regions and in marine ecosystems, as well as climate change and human health and others. So watch this space.
Yeah, personally, I appreciate very much this idea of forming NGOs. NGOs are very much so we will know very well what is the right person, what is the right person to do in I have personal experience of working with those in the countries and in other countries as well. I think NGOs obviously have a bit more insight how to deliver. Just posting one research gates scientific article without so much. The article needs to find the right mind of the right person.
For the most UN conference, must invite NGOs, major groups, youth, and main group, so participate. issue is very unique. So this is a part of the policy analysis. Very unique. We haven't given this an opportunity to comment on. So if she's still there and wants to say something, this is the opportunity to do so. Okay, so we're not going to clone Mark. It's a lovely applause. He's not such a lovely actor, but it's all very fun. Thank you all very much. It's been very helpful, I think, to us. I hope it's been helpful to you. Go out and change the world. Is I think what we're trying to ask you to think about doing. Encourage you to think about it. Before we stop running, there's a little presentation about NGO in this case. Somebody's working on air quality, and some of you will tell us very quickly about this. Then you're free.
Not quite, actually. Thank you so much, Roger. I really appreciate your time and opportunity to share what I'm going to share. Presentation. Okay. Okay, so I'm actually going to share this platform which we call VAXA. It's a Women in Air Quality in South Asia. It's a member-driven team, and it's so basically what we do. I'm just taking all the time to just share with you. So Pallavi Pant from Health Effect Institute, she's the founder, and I'm the co-team member, and along with other. Okay, on behalf of other VAXA member, I just wanted to share this platform so that you can also connect. And our basically the mission is to have a networking platform for all the women where we can encourage, highlight their work, and also have some opportunity to discuss and share what expertise they have. So it's not only the scientists, it's also all sectors involved in air quality. And so we have other. Okay, I will skip this, but we have cool online events. I mean, we did all of that. So I wanted to show the pictures. We have chai and chat just to, interact. Recently we had, and then we also something related to nitrogen. We had podcasts. So, we keep sharing that on Twitter and LinkedIn. And then we highlight some of our, women scientists or social scientist achievement there. We have mixers event. And we also have the social media page, Twitter, which is X, and then LinkedIn. So just connect there. If you have any question, please email. And, we would like to have you as a member. And, it's just to connect and share the expertise and, support each other. So thank you. Thank you for your time.
Before I give the floor over, so Professor Roger Jeffrey, first of all, thank you so much to all of the panel members. You did a fabulous job. Well done. And also a major thank you to Professor Roger Jeffrey. I don't know how many of you know him, but he is actually retiring at the end of this month. So I feel that today is probably his final, certainly in public, SANH moment. So I just wanted to give a round of applause to the fabulous work that Roger has done, and how literally in this group, in our SANH group, we managed to get Professor Raghuram, who wasn't too bothered about social policy, in all honesty, at the beginning, to at the end, at our last meeting in public, he actually stood up and told a story about how he got people socially engaged. So, and Roger, that's all down to you, so well done.
So moving fast, so I just would like to thank to all panelists for such a beautiful and wonderful session on the nitrogen policy recommendations and the growth around the globe. Now I just would like to request our chair, Professor Roger Jeffrey, just to honor our panelists. Just in 5 minutes, so I just would like to call first Liu to come and receive his token of appreciation. Then next is Mihai, that is from Romania. co-chair UNIP and working group. The next is Mihai from Romania, co-chair UNIP and working group. William St. Martin, Professor, WPI USA. Annie Yang, University of Edinburgh, UK. And lastly, Keisha from UK.
One more minute. So Keisha is also there.
Keisha is she's waiting for her. So to conclude the session and the day, I would like to thank you all for your patience and for your active participation in this event. I know everybody is very tired. So see you all tomorrow, and you all are kindly requested to join us for the dinner at the Hotel Legacy View. Thank you very much. Thank you everyone. Have a nice time.